Hello friends and welcome to CEC online lectures. In my previous lecture, I discussed about the microscopic evaluation of crude drugs, and in that we discussed about the chemomicroscopic evaluation and qualitative microscopic evaluation. And in qualitative microscopic evaluation, we discussed about the various microscopic structures that are used or that are studied under such kind of evaluation. Now, these microscopic characters or structures include different types of cells or tissues or trichomes, stomatas, or different ergastic cell contents. Now, in continuation with the previous lecture, today I'll start with the second part of microscopic evaluation of crude drugs. As I told you that in qualitative microscopic evaluation, we study different types of microscopic characters. Now, these microscopic characters can be studied either in a transfer section of the required plant part or through the powder microscopy if that plant part is provided in the powdered form. Now, I'll just go briefly through the methods by which we can prepare the sample or cut the transfer section and how we can prepare the sample for the powder microscopy. So first we'll start with the transfer section cutting techniques. Now broadly, we can cut transfer section with the help of two methods. Now I'm talking about the methods that can be used at the laboratory scale. The first method is when we cut the transfer section manually, that is the hand cut section cutting. And second is by using an equipment known as microtome. And when we use that equipment microtome, that process is known as microtomy. Now the sample preparation technique is different in both the methods. So I'll first talk about the hand cut sections or the manual section cutting. Now we can get a sample in two forms, either the fresh plant material or the dried plant material. So if we are getting the plant material in fresh form, then obviously we just have to keep the sample moist enough so that the cells or the tissues remain swelled as well as the sample remain soft. Whereas if we are getting the sample in the dried form, we all know that drying is done by evaporating the moisture, and once the moisture is evaporated from the sample, all the cells or the tissues, they get shrunk. So the first step of the sample preparation when you get a dry sample is to hydrate that sample. And now the hydration is done either by dipping the plant part or the required sample in the distilled water for sufficient period of time. Now if the sample is soft enough, say for example leaf is there, then simple dipping in distilled water or warm distilled water is enough. But if you are getting a hard sample, say for example you are getting a root sample or a bark sample or a stem sample, then simple dipping in the distilled water or the warm distilled water would not be enough. In such cases, we take the sample in the distilled water and boil it for sufficient period of time. Now, once the sample is softened or the tissues are hydrated, we hold the sample with the help of our thumb, index finger and the middle finger, and then we cut the sections with the help of a razor blade. Many a times it happens that the plant material or the sample material is thin enough or small enough to be held between the fingers. In such cases, we increase the surface area which we are holding, and then we cut the transfer section, or you can say we increase the surface area of the cutting surface. Now this is normally done by incorporating or embedding the required plant part or the plant material or the sample in a pith. And normally in laboratory scale, we use a block of potato as a pith. So we embed the plant material, say for example leaf or a small seed, which cannot be held between the fingers, and so they are embedded in the pith. Thereby we are increasing the cutting surface area, and then we cut the section with the help of razor blades. Now once the section are cut, they are transferred onto the slide. Then we add a drop of the required mountant, say glycerine in this case, and then we observe the characters of the microscopic tissues. Now coming on to the microtomy, or where we use microtome to cut the sections. Now the sample preparation technique in case of microtomy is altogether different or opposite to that what we saw in hand cut section or the manual section cutting. In case of microtomy, we have to first dehydrate our sample material by treating the sample material with different percentages of alcohol, and then we embed that plant material into a block of paraffin wax. Paraffin wax here acts as a pith, and then we cut the transfer section by placing that paraffin wax block having the plant tissue embedded in it into the microtome, and then we go for the section cutting. Now the major difference between the manual section cutting and microtomy is that we can select the thickness, or we can adjust the thickness of the section according to our required in case of microtomy. Whereas in case of manual section cutting, we cannot select the thickness, we cannot adjust the thickness of the section, transfer section. Second difference is in the sample preparation, which I have already discussed, and third is the post section cutting procedures. In case of manual section cutting, we just took the cut section onto a slide and put a drop of glycerine, and then we observed the tissue or the transfer section. In case of microtomy, after cutting the sections, we first have to de-wax the transfer section. Now de-waxing itself is a multi-step procedure, and I will not go into detail of those de-waxing steps. So after cutting the sections, in case of microtomy, we just de-wax the sample, and then we treat it with different reagents and mount it in a suitable reagent, and then we observe the sample. Further, staining can be done in both the cases, manual as well as microtomy, if required, to observe the other characters or the chemical characters of different microscopic tissues or cells. Now another advantage of microtomy is we get uniform thickened sections in case of microtome. This means that the whatever sections we are cutting with the help of microtome, they will have a uniform thickness throughout the transfer section. Whereas in case of manual section cutting, many times it happens the thickness of the section is not uniform throughout the transfer section. At some areas it will be thin, or at some areas it may be thick. So we sometimes we don't get a clear picture in a hand cut transfer section, whereas we get a very clear picture in case of microtomy. So this was how the samples or the section cutting is done in case of hand cut section as well as microtomy. So once the sections are cut, they are mounted on the slide, then we go for the observation of different characters. Now coming on to the sample preparation of a plant material when we get it in a powdered form. Now we all know that before we powder a material, we have to first dry it. And when we dry it, again the moisture is being removed from the plant material and all the tissues or the cells get shrunk. So the first step is to rehydrate or hydrate the powder or the cells that are present in the powder. And simultaneously, since some of the powders may have large amount of the coloring matter, say for example if we are getting a powder of a leafy plant material, or in case of some barks, they may have certain coloring agents. So second step is to decolorize that powder. Now in case of powder microscopy, hydration as well as decolorization or clearing of the sample goes simultaneously, and for that we require certain reagents which we call them as clearing or decolorizing agents. These are chloral hydrate, lactochloral, lactophenol, or sodium hypochlorite. Now amongst these, chloral hydrate is the most widely used decolorizing or clearing agent, and sodium hypochlorite is normally also used to decolorize certain deeply colored transfer sections. Say for example, if you are cutting a section of a leaf and it is highly pigmented, we can use sodium hypochlorite to decolorize. Obviously we can also use chloral hydrate, but sodium hypochlorite is largely used for decolorizing the transfer section. So once the powder sample is decolorized. Now decolorization is done by adding 2-3 drops of clearing agent onto a slide. Then we add a pinch of powder with the help of dissecting needle, and we slightly warm the slide. Now since all these clearing agents are made in distilled water, so this also helps in the hydration of the powdered material or the powdered tissues. So we do go for the gradual heating and addition, subsequently adding more and more clearing agent if required. Now this addition of clearing agent and heating, these steps are repeated till we get sufficiently cleared the sample material. Once the sample material is cleared, we add a drop of glycerine, put a cover slip, and then observe various tissues in a powder. So if staining is required, we can also do staining with different reagents and then observe the tissues. So this is how different microscopic characters are studied in a transverse section as well as in a powder microscopy. Now in this slide, you can see different characters that are studied in different plant parts, microscopic characters in different plant parts. Say for example, if we are getting a leafy drug, then we studied what kind of trichomes it is having, whether trichomes are unicellular or multicellular, covering or glandular. Then we also go for the characters of epidermal cells, whether their cell walls are straight or they are having wavy cell walls, or the epidermis is covered with cuticle, and if it is covered with cuticle, whether the cuticle covering is smooth or it is striated. Then we study about the palisade cells, whether they are single layered or multi-layered. Then what type of vascular tissues are present or vascular bundles are present, and what type of stomata is present in that particular leafy drug, and different types of ergastic cell contents such as calcium oxalate crystals or calcium carbonate crystals or starch if it is present in the sample. Then in bark drug, we study about the characters of the cork cells, whether they are thin-walled, thick-walled, single-layered, multi-layered, or if epidermis is present, what are the character of the epidermal cells. Then we study about the characters of the cortex, what kind of tissues are present in the cortex, whether it is parenchymatous or collenchymatous, whether the cortex cells are having any kind of cell inclusions or resinous material is present, whether they are having any kind of oil glands or fibers are present in the cortex or not. So all these characters for the cortex we observe. Then we study the characters of phloem fibers, what kind of shape is there, whether they are having pits on the surface or not. If they are having pits, whether those pits are bordered or normal pits. So these characters we study in phloem fibers. Then again we study about different types of ergastic cell contents, what kind of cell contents are present, what is their shape, structure. Then obviously sclerids and stone cells. Then in case of stem or the root drug, again we study the characters of the cork cells or epiblem. Epiblem is nothing but when the epidermis is present in root, we call it as epiblem. So we study the characters of epiblem, cork cells, cortex, phloem fibers, xylem vessels, what kind of secondary thickenings are present in the xylem vessels, or what kind of pits are present on the xylem vessels. Then we study characters of xylem parenchyma, whether the parenchymas are having pits on it, whether they are having enough starch grains in it, whether the walls of the xylem parenchyma are thickened or not. So these characters. Then we study about, we look for the characters of pericyclic fibers if present, then medullary rays, ergastic cell contents, sclerids or stone cells. And if we are getting a seed drug, then we look for the characters of testa, that is the outermost covering of the seed, then trichomes if present on the seed or not, then what type of hilum is present, what are the characters of the hilum, then endosperm, and then sclerids if present in the seed. So if we get a fruit drug, then we study the characters of epicarp, mesocarp, endocarp, what kind of cells are present in endocarp, what are the cell inclusions in the endocarp cells or not, or in mesocarp, then what kind of tissues, other tissues are present in the fruit drug, or whether the fruit drug is having any kind of sclerids or stone cells in it. So these are the different qualitative microscopic characters that are being studied in different plant parts. So now we move on to how these microscopic characters, qualitative microscopic characters, help in differentiating the drug from its adulterant or its identification. Now if you look into this slide, it has three pictures. First is of digitalis purpurea, second is verbascum thapsus, and the third is primula vulgaris. Now we all know that digitalis purpurea is a well-known cardiotonic drug, and the other two, that is verbascum thapsus and primula vulgaris, are its adulterants. Now just look into the picture and just imagine I give you a sample of dried leaves of Digitalis purpurea that are mixed with dried leaves of Verbascum thapsus or Primula vulgaris. So it would be very difficult to just uh, differentiate between the leaves of Purpurea or Verbascum thapsus or Primula vulgaris in the dried form by morphological evaluation, because all the leaves shown in this picture after drying, they will look morphologically similar. So it would be very difficult to differentiate whether the Digitalis has been adulterated or not with these two plants mere by morphological evaluation. So in such cases, we go for microscopic evaluation. It could be either with the help of transfer section or with the help of powder microscopy. So in this slide, you can clearly see that how the microscopic characters help in differentiating between the Digitalis purpurea from its adulterants. First is the stomata, and you can see stomata is anomocytic in all the three plants. Then are the covering trichomes. In Digitalis purpurea, the covering trichomes are uniceriate, multicellular, and they are 3 to 4 cell in length. Whereas they also have certain collapsed cells, and that is why they are also known as collapsed trichomes. In case of Verbascum thapsus, these covering trichomes are branched and they are known as candelabra trichomes. So this is how you can see the difference between the two purpurea and its adulterant Verbascum thapsus. And in case of Primula vulgaris, again the covering trichomes are uniceriate and multicellular, but the length of the trichomes in Primula vulgaris is 6 to 9 cell. So if the sample of Digitalis purpurea is having covering trichomes longer than the 6 cell length, we can say it may be adulterated with Primula vulgaris, or if it is having branched trichomes, we can say that it may be adulterated with the Verbascum thapsus. Next is the glandular trichomes. In case of purpurea, we are having unicellular glandular trichomes with unicellular stalk and a bicellular head, and rarely they have a unicellular head. Whereas in case of Verbascum thapsus, these glandular trichomes are not observed, while in case of Primula vulgaris, the glandular trichomes mostly or all the glandular trichomes, they have unicellular stalk and a unicellular head. It means bicellular head is completely absent in case of Primula vulgaris. So if we are observing large amount of glandular trichome with unicellular head in case of Digitalis, then we can say that Digitalis purpurea leaves may be adulterated with Primula vulgaris. So this is how these qualitative microscopic characters help in differentiating. So there is another example for leafy drug. I have taken Melia azadirac, that is Bachiana neem, and Azadiracta indica, which is common neem. So again, you can just imagine if the leaves of these two neem varieties are mixed together, it would be very difficult to differentiate mere by the morphological characters. So in this slide, you can see how the microscopic characters differentiate between the two leafy drugs. First is the epidermal cells are having straight anticlinal walls in case of neem, whereas Bachiana neem is having wavy walls in case of epidermis. Trichomes in case of neem are simple, covering but warty, whereas these covering trichomes in case of Bachiana neem are star shaped, and it also has glandular trichomes which are absent in case of common neem. Stomata is anomocytic and actinocytic in case of neem, whereas it is only anomocytic type in case of Bachiana neem. And normal common neem, that is Azadiracta indica, is having prismatic calcium oxalate crystals, whereas Bachiana neem is having needle shaped calcium oxalate crystals. Now in this slide, you can see three different trees, and the bark of these three trees are being used. First is Ficus bengalensis, second is Ficus religiosa, third is Ficus racemosa. So all three species are of the Ficus genus, and bengalensis is commonly known as bargat or the banyan tree, Ficus religiosa is commonly known as peepal, and Ficus racemosa is known as gular or the cluster fig tree. Now the bark of these three trees can also be differentiated on the basis of qualitative microscopy. As we can see in the slide, the stone cells present in the bargat tree are small circular or oval, and they are present in the isolated form or in groups, whereas the stone cells are small, triangular to ovate in case of peepal tree, and they are small, polygonal or squarish. Again, they are in the isolated or in the group form in case of gular tree. Similarly, sclerids, they are few, rectangular to oval in case of bargad, whereas in case of peepal tree, these sclerids are abundant, rectangular to irregular in shape, whereas they are completely absent in case of gular tree. And in case of cork cells, bargad is having polygonal to pentagonal or hexagonal cork cells, whereas they are polygonal to thick walled in case of peepal tree, and it is polygonal to hexagonal in case of gular tree. So this is how the qualitative microscopic characters also help in differentiating the bark drugs. Next in the slide, you can see the picture of two different root drugs. One is Rauwolfia serpentina and second is Rauwolfia vomitaria. Now, Rauwolfia serpentina is known as Indian Rauwolfia, and vomitoria is known as the African Rauwolfia. Now, if we look into the qualitative microscopic characters of these two root drugs on the slide, majority of the microscopic characters are same. So we can also say these qualitative microscopic characters also help in differentiating not only the morphologically similar drug but also microscopically similar drugs. So you can see in this table, Indian Rauwolfia is having 3 to 4 layers of thin-walled cork cells, whereas African Rauwolfia is having only 2 to 3 layers of thin-walled cork cells. Though the difference is there, but it's very minor. In case of Indian Rauwolfia, it is 3 to 4 layers, whereas in case of African Rauwolfia, it is 2 to 3 layers. Pericyclic fibers in both the cases are large, occasional and non-lignified. And again, xylem vessels are same in both the root drugs, that is, they are lignified and have bordered pits. Now, the difference occurs in case of sclerids. In case of Indian Rauwolfia, sclerids are completely absent, whereas in case of African Rauwolfia, sclerids are numerous. They are isodiametric to elongated rectangular in shape. So if we are getting a sample of powdered sample of Indian Rauwolfia, and if we observe sclerids in it, we can definitely say that the Rauwolfia sample, with the powder Rauwolfia sample which we have obtained, either it is not pure or it has been mixed with the Rauwolfia vomitoria or any other powder of the plant material or powder material of the another plant in which sclerids were present. So presence of sclerids will clearly show that the powder sample of Rauwolfia that you have obtained is not pure. And again, the calcium oxalate crystals in both the cases are same, that is numerous and prismatic. So friends, in this lecture we discussed how the qualitative microscopic characters are studied in transverse section and powder microscopy, how the sample is prepared in case of transverse section by manual or hand section cutting, as well as in case of microtomy, how the sample is prepared in case of powdered material, and we also took various examples to understand how these qualitative microscopic characters help in differentiating between the adulterants or identification of a particular drug. So friends, in the next series of the microscopic evaluation drug, I'll be starting with the quantitative microscopic aspects, and in that we will discuss different types of quantitative microscopic techniques. Thank you.